Last time, we talked about non-human aging system, especially C. elegans, Drosophila melanogaster, and the mouse model. But the best system to understand human aging is, of course, the human system. For all the advantages these model systems that we talk about have, they all suffer from the same shortcoming. They are not humans. Humans are also the best understood organism when it comes to aging. We have a very clear understanding of aging phenotypes, progression of aging. We have mountains of data on that. We know the causes of death and the diseases that are associated with old age. However, there's one drawback to human studies. For ethical reasons, we can't really experiment in humans, thus human studies are largely restricted to observational studies and not interventional studies. But even so, we can get a lot of information from human studies. Human population growth took off at the turn of the 20th century, at the tail end of the Industrial Revolution. These increased population numbers go hand in hand with increased life expectancy. Interestingly, the biggest factor in these advances of population growth, and especially for our context, life expectancy, is most likely the dramatic reduction in infant mortality. Once an infant successfully has made it out of childhood, even when centuries passed, it could expect to live a fairly long life. Thus, life expectancy at birth is really not a very good yardstick to measure longevity. Archaeological evidence actually shows that once an individual had survived the perilous infant years, average lifespan was not too dissimilar from modern days. Even Neanderthals, more than 56,000 years ago, were not particularly short-lived. As one of the best preserved skeletal specimens, the famous Old Man of La Chapelle demonstrates. Thus, advances in longevity are largely due to better nutrition, hygiene, advances in medicine, and not because we learned how to delay aging. In a sense, our increased life expectancies are due to the work of Antony van Leeuwenhoek and Louis Pasteur, who essentially fathered the field of microbiology, which then in turn led to the discovery of disease-causing germs and, even more importantly, the treatment of these disease-causing germs. In terms of our mortality rate curves that we discussed earlier, this increased life expectancy is therefore due to a lowering of the y-intercept, or the hazard function, and not because we interfered with the slope of the Gompertz curve, the actual rate of aging. In terms of aging, humans are very special in that they are exceptionally long-lived for the size and metabolic rate, making studies in humans fairly complex and very difficult to carry out. One approach to elucidate the aging process in humans is to investigate diseases of accelerated aging, so-called progerias. We already talked about the best-known progeria, Werner syndrome, which is due to a mutation in the enzyme Werner helicase, an enzyme that is involved in the unwinding of DNA and repair of DNA damage. Werner's patients exhibit many signs of aging, such as arteriosclerosis, type 2 diabetes, skin atrophy, hair loss, graying, osteoporosis, cataracts, and then, of course, shortened lifespans. However, they may be remarkable in other organ systems. On a cellular level, patients show increased cellular senescence, which may account for the tissue aging phenotypes. Organs may be unable to self-renew and unable to repair damaged cells. However, Werner syndrome is still only a disease that shows signs of accelerated aging in the affected organ systems rather than a full-blown accelerated aging of the entire organism. The usefulness of progerias in studying the aging process is therefore quite limited. Another way to study aging in humans is by performing twin studies. Examining identical twins, researchers can uncover markers of human aging to estimate the influence of the environment on the aging process. Using this approach, it has been estimated that aging is governed by genetic factors by approximately only 25%. Even NASA has gotten into the aging business. These twin astronauts, Scott and Mark Kelly, are being studied for the effects of space travel on aging. Unfortunately for Mark, he was chosen as the Earthbound control twin, while his brother Scott spent a whole year at the ISS. A paper just came out in early 2018 that described the genetic and gene expression changes that Scott experienced while spending a year in space. Probably the most powerful way to study aging is to study centenarians, or even so-called supercentenarians. These are exceptionally long-lived individuals, 100 years and older. These populations are exceedingly rare and especially enriched for individuals that are extremely resistant to diseases of aging. These supercentenarians are responsible for the slowing down and the decrease in the age-specific mortality rate at extreme old age that we can see in the Gompertz curve, like we discussed earlier. Using these populations, researchers are now able to study a whole host of morphological, physiological, and molecular phenotypes. Human aging phenotypes usually manifest themselves in a whole range of diseases, onset of which is very tightly correlated with old age. Especially supercentenarians show a remarkable resistance to many of these diseases and often do not experience them at all. When the correlation is tight, we can use the disease/observation as a so-called biomarker of aging. With an ideal biomarker, one would be able to only measure the marker itself. We call that the physiological age, and then extrapolate to the chronological age of the individual. Appearance of such a biomarker of aging would be delayed in an older individual and accelerated in a short-lived individual. In other words, one may be able to predict remaining years of life by measuring the level of such a biomarker. Many potential biomarkers of aging are listed in this table, and they range from blood pressure to body mass index, the level of cholesterol in your blood, fasting glucose levels, and especially gait speed or grip strength. Moreover, human aging studies can also be done in a longitudinal way. That is, by following individuals and families through the years and tracking these biomarkers of aging. Using this approach, it was discovered that extreme longevity in fact often does run in families, stressing the importance of genetic factors to the aging process. With the deciphering of the human genome, it became feasible to search for these genetic factors that influence human longevity. With modern genetic tools, it is now possible to detect the difference in a single DNA nucleotide at a particular genetic locus. These single nucleotide polymorphisms, or SNPs or SNPs, are responsible for the phenotypic variation between individuals. Thus, some SNPs may be associated with extreme longevity, and thus identification of these longevity SNPs will be able to tell us more about the genes and the mechanisms involved in the aging process. Similarly, CNVs, or copy number variations, the number of copies of a particular genetic marker in an individual's genome, may also be used for longevity studies. Modern genomic tools allow to take SNP analysis to the next level and to perform data mining deep into the human genome. In linkage studies, whole groups of families are investigators for biomarkers associated with disease phenotypes, or in our case, longevity. These studies can reveal whole chromosomal regions that are associated or linked to that particular phenotype, not only in a single family, but in several, which increases the likelihood that genes contained within that chromosomal region are important for that particular phenotype. A related approach is called genome-wide association study, or GWAS, that looks at hundreds of thousands of SNPs simultaneously to detect any SNP associated with a particular trait. These kinds of approaches are especially pertinent for aging research, as aging is a highly complex trait that is multigenic in nature and not simply caused by a single gene. Once a number of SNPs associated with a trait has been identified, they can be grouped together into a so-called haplotype, a group of SNPs that is always inherited together. These SNPs, or haplotypes, thus usually occur together on the same region of a chromosome. Thus, aging-associated haplotypes can be identified and mapped, and that is then followed by the elucidation of the genes involved in these haplotypes. Figure 6.6 shows the typical workflow of such an experiment. GWAS was used to identify a chromosomal region tightly associated with a trait. Analysis of that region, as shown in panel B, shows that it contains two protein-coding genes. In this case, they are inhibitors of CDKs or cyclin-dependent kinases. These are enzymes that play a very important role in the control of the cell cycle. And one non-protein coding gene. In fact, this is a CDK inhibitor. Individual SNPs can now be mapped directly onto the genetic data to show all the different SNPs associated with disease phenotypes in these genes alone. Once a candidate has been discovered, it needs to be shown experimentally that it indeed may influence aging and longevity, which is best done in non-human model systems. Using modern genetic tools, a researcher can now overactivate such a gene, underactivate, or even completely disable it. Activate it only at certain times of life, or only in certain organs, only in certain tissues, and then determine the effects of this intervention on aging phenotypes and longevity. The researcher can even go further and genetically engineer this gene to potentiate its effect. Ultimately, one would then develop small drugs that may mimic the effects of this gene in order to develop a pharmacological intervention that may be able to ameliorate, at the very least, the detrimental effects of the aging process. But we will talk about that more in the final section of this course. Ever since aging research took off in the mid-80s, we have made great strides in identifying such candidate genes, and we will encounter them throughout this course. We know, for example, that ApoE, or apolipoprotein E, a gene involved in the development of Alzheimer's disease, also affects the aging process. Genes involved in TOR signaling, a pathway that is involved in the quality control of proteins, affect aging and longevity. And likewise do genes of the insulin-IGF pathway, a metabolic pathway that affects metabolism and the development of type 2 diabetes. And so do sirtuins, enzymes that are involved in metabolic regulation. Finally, as mentioned earlier, the genetic component of human longevity is estimated to be around 25%, which means that a good 3/4 of human longevity can be attributed to other factors, and most importantly, is not transmitted genetically. The environment can interact with the genome by creating certain markers on the genome, such as methyl groups or acetyl groups, on DNA and histones, the proteins that hold the DNA together, which in turn can regulate the activity of genes. Markers like that are called epigenetic markers, markers that are outside the actual DNA sequence. Thus, the epigenome, the totality of all epigenetic markers on DNA and histones, can greatly affect longevity and aging. Deciphering the human epigenome and how that influences aging is truly the next big step in aging research. Check your understanding. Define human aging phenotypes. Compare and contrast different experimental systems for studying human aging. Explain how to link longevity phenotypes to genes.